Hello, this is a presentation being presented by Stephanie Davis and Mark Kennemeth. It involves Biology 1, and what we're going to get into in this presentation is bulk transport. There are different types of bulk transport that we'll get into in this video. The first would include exocytosis, the second endocytosis, which within that there's three other different types of endocytosis transport. The first being phagocytosis, second pinocytosis, and C receptor mediated endocytosis.
Okay, the first form of bulk transport we're going to talk about is exocytosis. Exocytosis is taking something from inside the cell to outside the cell. What we're taking out can be anything from waste, debris, just things the cell doesn't need anymore, and it can also be nutrients for another cell, or maybe proteins or a hormone, anything, taking it from inside to outside. Exocytosis is literally the reverse of endocytosis, which we'll get into endocytosis a little later on. So what happens is we have a cell wall. It's our cell wall. This is the outside cell, inside the cell. We have a Golgi apparatus right here. And what happens is a transport vesicle that has budded off from the Golgi, so this is our little transport vesicle budding off, moves along microtubules of the cytoskeleton to the plasma membrane. So it's moving along, goes out here. When the vesicle membrane and the plasma membrane come into contact, specific proteins rearrange the lipid molecules of the two bilayers so that the two membranes fuse. So this is what happens. They fuse together like this. The contents of the vesicle then spill to the outside of the cell, and the vesicle membrane becomes part of the plasma membrane. So then this right here will open up, and everything that we have inside will go to the outside of the cell. And like I said earlier, this can either go to another cell, or it could be waste and it's just going to get flushed out. Whatever it may be, it's coming to the outside of the cell. Then this right here will fuse with the cell wall right here, the membrane. This will fuse with the outside of the cell. When this fuses with the outside, it helps regenerate it, because when we do endocytosis, we're taking away from our membrane here and bringing things into the cell. So when we do exocytosis, we're adding back to it. So it's sort of like a recycling phase. Many cells in the body do this. Secretory cells use this to export products, like I said, to other parts of the body. Some good examples are fungi. Fungus do this because they produce a chemical inside of their bodies and they excrete it on the outside. The reason they do this is so that other creatures won't eat them or consume them, so they can live longer. Another example of this is the pancreas. The pancreas does this with insulin. It produces the insulin and secretes it out to be used throughout the cell to regulate our blood sugar. It excretes into the extracellular fluid. Another example of this would be digestive enzymes in the digestive tract. Also, any type of adrenal gland or hormone gland or mucus-producing gland will also do this right here.
Now, in this form of endocytosis, we're going to discuss phagocytosis, and essentially it means cell eating. The cell engulfs debris, bacteria, or other sizable objects. Phagocytosis occurs in a specialized cell called phagocytes, include macrophages, neutrophils, and other white blood cells. And we have our cell wall here, and essentially what it does is that they term it invagination, where it opens up. And then it takes in the particles into it and pinches off into a vacuole. So it has your food particle inside here. And a lot of times it'll fuse with one or more lysosomes containing hydrolytic enzymes. Materials within the vacuole are broken down and degraded. A cell engulfs a particle by wrapping around the pseudopodia, which is right here, kind of like what I just went over. That's what they term that portion, and packaging it within the membranes. This is called a food vacuole. The particle will be digested after the food vacuole fuses with a lysosome containing hydrolytic enzymes.
Okay, we're going to talk about penocytosis. Penocytosis is another form of endocytosis. But in penocytosis, we're consuming fluids, not solids, such as in phagocytosis. Penocytosis literally means cell drinking. So we're going to be taking in fluids. So we have a cell wall, and there are little solutes that float around in the extracellular fluid outside the cell. Our cell wants to get these solutes inside of the cell for different purposes. So what our cell will do, it'll pinch off and consume some of these, but it's not wanting to consume the fluid necessarily. It's wanting to consume the solutes that are dissolved in the fluid. That's where we get the name cell drinking or penocytosis. The cell will consume, once it gets all the solutes that it wants, it'll close off. Once it closes off, it'll make a vacuole. This vacuole will then, once it's consumed all the nutrients it wants, enter the cell. The cell can then disperse these to wherever it needs, to whatever needs to be done in the cell. It can also help break down these solutes even more once it fuses with a vacuole. Penocytosis is nonspecific, meaning it'll consume most anything. It doesn't look for a particular enzyme to make this reaction possible. Okay, the last one we're going to learn about is receptor-mediated endocytosis. Unlike penocytosis and phagocytosis, this one is very specific on what it will allow into the cell. In receptor-mediated endocytosis, we start out with the cell wall. This is the outside of the cell, this is the inside of the cell. The outside of the cell, we have little receptor sites all over the cell on the outside. And then we have different things floating around outside the cell, such as proteins. A good example of this is how the body takes in iron. It has to have these for the body to absorb iron, these little sites. The body will then recognize the iron and allow it into the cell. Another example is cholesterol. But what the sites do is these little proteins or whatever molecules are floating around outside the cell. They then will float down and connect with the receptor sites. So they plug in right here, connect with the receptor sites. Then once that happens, the cell will start to dip, pinch off in a way like it did in previous examples. So that's it, dipping in and pinching off. But the cell doesn't necessarily want these proteins. It might need these proteins for something inside the cell, but it doesn't necessarily need them. There will be other solutes floating around that the cell knows that when these proteins are present, these little solutes will be present also. So it could be these solutes that the cell needs more than these proteins that are bound to the receptor sites. So once these receptor sites are all full and it knows these little solutes are floating around in there, the cell will begin to pinch off, and eventually it'll pinch completely off. I'll just draw a few of them. Eventually it'll pinch completely off and be inside the cell. At this point, a vacuole can attach to it and inject its digestive enzymes, or a lysosome can attach to it and inject its digestive enzymes, digesting this even farther. That way, the cell can then use this for whatever is needed, food or energy, whatever need be. Like I said earlier, this is responsible for the uptake of iron in humans as well as cholesterol.